Our next speaker is Professor David Withers from the University of Birmingham in Birmingham, England. David, I've given you permission to start sharing your screen. Excellent, we've got that pulled up. Professor Withers qualified with a Bachelor's of Science in Virology and Microbiology from the University of Warwick in 2000. He went on to study for a PhD in immunology at the Institute for Animal Health in conjunction with the University of Bristol. After obtaining his PhD, he continued his studies in the laboratory of 𝘷𝘪𝘷𝘰 at the NIH. He then returned to the UK to study with Peter Lane at the University of Birmingham, cementing his interest in the secondary lymphoid tissue development, structure, and how this controlled CD4 T cell responses. Research in the Withers Lab is supported by the Wellcome Trust. In 2011, he was awarded a Wellcome Trust Research Career Development Fellowship to establish his own research group investigating the role of lymphoid tissue inducer cells in lymph nodes. In 2016, he was awarded a Wellcome Trust Senior Research Fellowship in Basic Biomedical Science, and he'll be sharing with us how they've tracked dynamic changes in tumor infiltrating lymphocytes in vivo. Professor Withers.
Thank you, Emily, and thank you to ProImmune for inviting me to talk today. So what I'd like to do is really share some, I think, quite basic but fundamental novel data where we have been trying to understand the movement of cells into tumors and how they change over time. So we all know that the lymphocytes that we see in tumors look different. It's well understood that for CD8 T cells, the constant exposure to antigen causes these cells to become exhausted. And there's been a lot of work unveiling this phenotype and how this is driven. We also know that other effector lymphocytes, such as NK cells within the tumor, also lose key functions. There have been interesting studies describing these as becoming more ILC1-like. And there are also a whole bunch of other immunosuppressive mechanisms that we know operate within tumors. However, if we think about how cells are changing within a tumor, I think there are some very basic questions that we still don't really understand the answer to. So one would be, what are the phenotype of the cells that actually enter the tumor? How heterogeneous are these populations? And where have these cells actually come from? And a further very basic question is, well, do any cells actually leave a tumor? If they do, where do they end up and what do they look like? So why is unpicking these kind of questions interesting? Well, obviously, as we heard from the last speaker, checkpoint blockade immunotherapy has revolutionized our understanding of how to treat cancers, but there is still a lot of room for improvement. And if we better understood how cells change within the tumor, I think it's reasonable to anticipate that we could refine interventions and coordinate therapies better. And ultimately, this should lead to enhanced successes in the clinic. To come back to the specific dynamic questions that I posed, a further point would be resolving this question about bystander CD8 T cells that have been described within tumors. But to come back to the dynamics, if we want to understand how to refine some of these therapies, we should understand where these therapies are operating, how much of the effects of immune checkpoint blockade are operating within the draining lymph node, and are there other ways that we might combine with this to enhance anti-tumor responses by targeting draining lymphoid tissues. We also know that the systemic response to tumors is important, and there's been some stellar recent publications really underlining the importance of T cell expansion outside the tumor in predicting responsiveness to checkpoint blockade. If we understood where the cells leave, this might also open up new ways of monitoring anti-tumor responses. At the moment, we're very bad at understanding whether a therapy that's being employed in patients is actually working very well. So how did we come to this area of work? Well, my lab for a number of years has been interested in how you regulate T cell responses. And through those studies, we became interested in trying to understand the movement of cells in vivo. So we acquired a fantastic transgenic mouse model that was made many years ago now by Michio Tamura. And this is called the Kaede Photoconvertible mouse. So this is a transgenic mouse where all cells in the mouse express a green fluorescent protein. And upon exposure to violet light, this green protein irreversibly switches to a red form. So what we've been doing in my lab to look at the movement of cells in and out of lymph nodes was to set up an approach by which we could, through the exposure of light, turn all of the cells within a lymph node from green to red. So our strategy here was if we could label all the cells in a lymph node red, we would be able to have two very basic but clear parameters of new cells entering. These would have the endogenous green fluorescent version of the protein. We'd also be able to see cells leaving the lymph node, as these would have the converted red version. We'd also be able to see the retention of cells within the tissue as red cells that retained within the lymph node. So this simple process has enabled us to look very carefully at the movement of T cells and innate lymphoid cell populations in and out of lymph nodes. But it was from here that we really became interested in trying to understand whether we could apply this technology to better understanding the movement of cells in and out of tumors. So our approach here was to use a subcutaneous tumor cell line model. Obviously, we've been photoconverting or labeling lymph nodes, so it wasn't such a big leap to try and label a tumor growing subcutaneously on the flank of one of these Kaede mice. So the tumor model I'm going to show you here is an MC38 colorectal cell line that many of you will be familiar with. We've implanted this on the flank of the mouse, and then we have exposed it to violet light through the skin to label the cells. So when in all the data I show you on this slide, we're going to look at two simple parameters, whether the cells have this red fluorescent protein version, and we're saying this means that the cells have been retained in the tumor since prior to labeling, or whether the cells occupy this space here, and we think these cells are newly entering the tumor after labeling. So to show you some controls, so this is a tumor that has never been labeled, and you can see all of the hematopoietic cells labeled with this green protein. If you then look at the cells immediately after photoconversion, you can see we've turned all of the cells red. If we look in the draining lymph node, you can see that the cells are still green, suggesting that our labeling is specific to the tumor and it's not hitting surrounding tissue. If we then look at different cell types within the tumor, here I'm just showing you different types of tumor infiltrating lymphocytes, so CD8, CD4s, Tregs based on FOXP3 expression and NK cells. You can clearly see that when we look at different time points, we can see clear red populations and distinct green populations. So just focusing at 24 hours, if we look at CD8 T cells, about 3/4 of the CD8 T cells in the tumor are red, but you can see that 1/4 of the cells are clearly distinct with this, and expresses green version of the protein. So we can track changes over time and look in different cell populations. If we enumerate this across many experiments, you can see that the proportion of green lymphocytes, regardless of their type, is increasing substantially over time. If we then look at total numbers, so I draw your attention to this as a logarithmic scale, you can see that actually the number of red TILs within the tumor is staying relatively constant. However, if you then look at the number of green cells in the tumor, you can see, again, a logarithmic scale, a fairly substantial increase in the number of cells entering the tumor over this 5-day period of time. So one of the concerns about this kind of approach is that proliferation will result in the loss of this red label. To just show some evidence that we think this isn't a problem within our system, if you compare the level of proliferation based on Ki67 expression in the draining lymph node or the tumor, you can see that while T cells in the draining lymph node show high levels of Ki67 expression, we detect very little within the tumor. If we look at Ki67 expression versus Kaede red, you can see that there's no evidence of a Ki67 high, Kaede red low, or negative population emerging over time. So we don't think there's an enormous problem with proliferation, and we think, as others have described in tumors, if you look within lymphocytes, a key point is that the T cells are not proliferating a great deal. And you can see if we look at another marker, we see interesting changes that I will come back to shortly. So what I've shown you so far is a very kind of overview of the lymphocyte populations, and we really wanted to fully capture the heterogeneity in the populations to the best of our ability. And the strategy we've used for this is to use single cell RNA sequencing. So it's the same experimental setup. We've labeled a tumor on the flank of the Kaede black 6 mice, but this time using FACS, we've isolated the green and red populations at 2 different time points, giving us 4 samples in which to look at by droplet-based single cell RNA-seq using the 10x platform. So just to very quickly show you the kind of data that everyone in the world is now quite familiar with, we've looked at all of the clusters within our populations. We can broadly see the expected TIL populations we've described. We've then split these further into clusters based on the normal process of looking for expression of lineage-defining genes. A really interesting and very basic point at this stage is we can obviously ask whether the cells came from the green or red samples that we isolated and which time point. And if we simply look at the different types of cells, you can see it clearly for NK cells here, you can see that whether we compare those cells from the green or red samples, they actually segregate quite distinctly within these clusters, suggesting that there are quite distinct transcriptomic differences between the cells based on their time within the tumor. So I'm now going to focus in on the CD8 T cells. So this is just looking at the clusters of CD8 T cells within the tumor. And then this is showing those clusters and whether the cells came from the different samples. So 24-hour green cells, 72-hour green cells, 24-hour red cells, and 72-hour red cells. And the key point is that you can see that depending on which sample you look at, they occupy different clusters. These pinwheels are really showing the proportion of each cluster that is dominated by green or red cells. So we have two clusters that are really dominated by the newly entering green cells, which again you can visualize here. And the remaining three clusters are really reflecting cells that have been in the tumor for longer. So we obviously done lots of biased and unbiased analyses of this and tried to pull out ways of better understanding what these different populations are. And one striking initial observation was if we look at our cluster 1, it basically had a whole bunch of markers that look an awful lot like a naive T cell. So is this really true? Do we see significant numbers of naive CD8 T cells entering our tumor? So we went back to the flow cytometer. If we gate within the cells isolated from the tumor and we look at green and red CD8 T cells, yes, within the green CD8 T cells, you can see an enriched population of essentially naive CD8 T cells. If we then look at markers such as PD-1 and granzyme B, if you compare the total CD8s to the population of naive phenotype cells, you can see that they lack expression of these activation markers. So we think that this cluster 1 within our data is really, these are naive CD8 T cells that are freely entering into the tumor. So then an obvious point would be, well, if cluster 1 are naive CD8 T cells, what are these other cells entering the tumor within the first 24 hours? So to look better at this, we've tried to dissect the populations using antigen-specific reagents. And here we're very grateful to ProImmune for providing us with a pentamer that tracks an MC38 neo-antigen. In our hands, this pentamer works very nicely. And if we look within either the green or the red CD8 T cells within the tumor, we can see a discrete population of pentamer-specific cells. If we phenotype these cells, you can see that whether the cells came from the green or red populations, neither of them express a naive phenotype, and they both express high levels of 44, consistent with an activated phenotype. Interestingly, if we look at those cells in the green population, so these have been in the tumor for less time than the red populations, you can see heterogeneity in their expression of markers such as PD-1 and LAG3, whereas the red population are fairly homogeneous and express both. So to come back to the simple question I asked at the beginning, is there heterogeneity in the cells entering the tumor? We would say very clearly yes. Interestingly, we see naive CD8 T cells, and then we can also identify a distinct population that we think represent those that are responding to tumor neoantigens and have likely been primed in the draining lymphoid tissue and have now trafficked back to the tumor. So obviously one of the great joys of single cell RNA-seq data sets is just the volume of data you have. I'm not going to go into it in great detail now, but we can plot trajectories of how these cells change. We can route our trajectories in this population of cells that we think has newly entered the tumor, and then dissect some of the changes to really find granularity in terms of how they change over time. To provide just very simple proof of principle, again using the MC38 pentamer, you can see that if we track these cells at 24 or 72 hours and split between the red and the green cells always show less expression of markers such as PD-1 and LAG3 compared to those cells that are demonstrably been in the tumor for longer. So we think this approach will enable us to validate some of the interesting signatures that we see emerging from the single cell RNA-seq data. So I'm going to now switch to a different type of immune cell that obviously has a key role within the tumor. What if we look at the regulatory T cell populations? So within our single cell data set, there are 4 discrete clusters. Again, if you look at them overlaying the sample type with the clusters, we have one cluster up here, which is Treg cluster 1, that is really dominated by cells that have newly entered the tumor, these green cells, whereas those cells that have been in the tumor for longer really occupy these other 3 clusters, clusters 2, 3, and 4. And these are just pinwheels that are, again, showing the data in another way, asking the composition of the different clusters. So Treg cluster 1 is dominated by green cells. The other 3 clusters reflect populations that have been in the tumor for longer. So then an obvious point is to try and pull apart what these clusters are again. Again, this is showing an unbiased, this is the top 50 𝘷𝘪s. And from here, we identified some markers that we thought we could potentially pull apart these clusters. And then this was the task I set the guys in the lab. Can they pull apart these populations and really identify them at the cellular level using flow cytometry? So we picked these markers, neuropilin-1, LAG3, CD25, and IL-10. So this is now gating on Tregs within the tumor. This is looking at the red Tregs, so those that have been in the tumor for longer. And I think you can reasonably suggest that there are 4 populations of cells here. I think we have cells that resemble Treg cluster 1, putative Treg cluster 2, Treg cluster 3, and Treg cluster 4. What made this data really exciting was when we looked at the green Tregs, so those that have only just entered at 24 hours or have entered within 72 hours, you could clearly see the evolution of these cells. So if we look at 24 hours, the Tregs entering the tumor only have the phenotype that we think of clusters 1 and 2. Within 72 hours, you see some of these cells adopting a phenotype where they've upregulated LAG3 and CD25 and maybe a hint of IL-10. So what was amazing to us was how nicely this data recapitulated what we saw in the single cell RNA-seq. And we're now just validating these populations and checking some of the other markers, because if this gating strategy is correct, then we should be able to confirm some of the other things we see in our sequencing data. So a very simple question that I posed at the start was whether immune cells actually leave the tumor. So to ask this question, we have photoconverted the tumor and then looked for cells within the draining lymph node. So you just have to change your thinking about the colors for this type of analysis. So now, if we're looking at red cells, the red cells are those that have trafficked from the tumor and have moved to the tissue we're now looking at, whereas the green cells are essentially cells that were in the tissue and have probably never been anywhere near the tumor. So if we look at the different populations of red cells, we can see 2 main populations. One might expect to see dendritic cells leaving the tumor and pitching the draining lymph node. Indeed, we can see that there's a large population of DC within the cells that we see pitching up in the draining lymph node. But interestingly, we also see a substantial population of T cells as well. So to show you what the DC data looks like, so we've gated on DCs in this manner, and we've split them into CDC1 and CDC2. And what's very nice is that if you look in the draining lymph node, there are really discrete populations of Kaede, CDC1 or CDC2 that we think have left the tumor and have now migrated to the draining lymph node. Obviously, we're phenotyping these cells, and an interesting phenotype can be seen when you compare expression of PD-L1 and PD-L2 between these different populations. So I said we also saw T cells leaving the tumor. So this is now looking at the CD8 T cells arriving in the draining lymph node or the spleen. See, it's a discrete population on the flow cytometry plot. If we then phenotype these cells within the draining lymph node, we see a clear population of naive CD8 T cells as well as other populations. And we see this naive CD8 population also within the spleen. If we look at the Tregs, again, we can see these in the draining lymph node and making it to the spleen. And these only have a phenotype consistent with the cells that we actually saw moving into the tumor. So we only see what we were calling Treg clusters 1 and 2. So what I'm trying to say to you is that I think this data where we actually track what comes out of the tumor and compare it to what we see going in suggests that both naive CD8 T cells and thymically derived Tregs can enter, but they pass through the tumor and then exit again. And really the retention of cells within tumors is, I think, not completely surprisingly linked to the recognition of tumor neoantigens. It's actually the responsive cells that are retained within tumors, which is really what we see in our model. So one of the things we really want to get at is the contribution of the draining lymphoid tissue to what is arriving in the tumor, and to try and pull apart this idea of what comes from the draining lymph node versus potentially a peripheral memory cell pool. So in this kind of experimental setting, we've actually photoconverted the draining lymph node, and we're now looking for red cells moving back to the tumor. This is then what happens if we look in the draining lymph node 24 hours after photoconversion. We see the retention of a red population of cells within here. This is actually tracking a different antigen-specific population. We're using MC38 over here, and this is looking at over tetramers. We can see that there is a cohort of antigen-specific CD8 T cells in the draining lymph node, but when we actually look to where they go, we can track some back to the tumor. We can also track some moving out to the spleen, and you can see them here. So it's interesting that T cells can move from within 24 hours directly from the draining lymph node to the tumor, but the question is really whether there's any difference between the cells that go straight to the tumor or they move to the spleen, and we're currently trying to pull this apart. So to return to the diagram I showed you at the beginning, when we're trying to understand the changes that happen within the tumor, we can show that we have direct movement of cells from the draining lymph node, but we also have some cells that we think are forming part of a peripheral pool, and these then subsequently move into the tumor. And as I say, we're trying to pull apart the differences there. I think a key point about this movement of cells into the tumor is that it is heterogeneous. We think there are fairly relaxed rules on what can enter from the blood. And to come back to the question of bystanders, I think a lot of this could be explained by the kind of snapshot effect of looking in a tumor at a moment in time, or in using histological methods. Because clearly cells can enter, but I think many cells that don't show reactivity to tumor antigens promptly then leave again, which is kind of dynamic analysis can reveal. And as I've shown you, we can also track cells leaving the tumor, and we have several different populations that we're further phenotyping. So I guess my key points are that the retention of antigen-specific populations is really clear within this model, but it's striking how many cells seem to be able to enter the tumor and transfer through. So I will finish there. I'd just like to thank the people in my lab, my collaborators, and particularly guys in Manchester and in Cambridge, who provided phenomenal help with the bioinformatics analysis and the single cell RNA-seq. I'm very grateful to the creator of this fantastic mouse tool, and also my funders, particularly Wellcome AstraZeneca, Cancer Research UK, and recently the Cancer Research Institute. Thank you.